guys. Okay, so today we're going to be talking about something that has kind of a lot of relevance at the moment, given the entire COVID situation and the globally unique or this periodically unique time we're in. So today we're going to be talking about principles of disease and be having a bit of a look at epidemiology and pandemiology and all of that. So yeah, we're going to be focused on the pathology. We're going to be looking at diseases, and we're going to be looking at etiology. Okay, so first of all, let's start here with some principles of disease and epidemiology. So one thing that we should be and stay aware of here is that microbes are not all harmful to us. Actually, life on the planet would not be possible without microbes, but even on a smaller scale. So animals, which includes humans, are generally free of microbes in utero. However, at birth, normal and characteristic microbial populations begin to establish themselves, and they form a symbiotic relationship with us. So remember here that symbiosis is a close relationship between two species in which at least one of the species benefits. And within symbiosis, there are 5 main symbiotic relationships, which is mutualism, commensalism, predation, parasitism, and competition. And within our the relationships we share with our normal microbiota, we typically find commensalism, which is a biological interaction in which one of the members, in which members of one species gain benefit while the other member is not benefited or harmed. We also find mutualism, where both of the species gain benefit, has a net benefit. And we also find parasitism, where one organism, the parasite, lives in or inside another organism, the host, causing it some harm, and is adapted structurally to this way of life. But within ourselves, like I said, we have normal microbiota, which can benefit the host by preventing overgrowth of harmful microbes. So this is a form of inhibition, of competitive inhibition. And there can also be the elimination of these normal microbiota through antibiotic therapy. So if you drink too much antibiotics, or you don't adhere to the schedule, or you drink unnecessary antibiotics, you could potentially suppress your own naturally occurring microbiota, killing the bacteria specifically, which can result in some serious fungal infections. Some candidase infections is a good example of this, where you can see certain people growing some having fungal infections on their mouths and tongues and stuff like that. It appears almost like this white covering, and that's a function of the people drinking antibiotics, which kills their beneficial normal microbiota, allowing this fungus to grow where these bacteria would normally outcompete it, and then thus the fungus is able to grow where it normally is not able to grow. Okay, so again, these normal microbiota will permanently colonize those, so they're always there. But you can also find some transient microbiota that can be present for either days, weeks, or months. They are also notably, they're also normal microbiota, but they are just simply not permanent. And then just a definition there again of what this symbiosis or this relationship is. And in this case, the symbiosis is specifically referring to the relationship between the normal microbiota and the host, so in this case, us. So we're going to be continuing on here, looking at some examples of symbiotic relationships. So if you look at an example of commensalism, this can be a example of the corneobacteria in the eye, where the microbe benefits, but we are unaffected by its presence. Mutualism, where both parties benefit. So normally occurring E. coli that will synthesize B vitamins and K vitamins in the lower GI tract, and that's the gastrointestinal tract. But this can shift over to parasitism, where the microbe benefits and the host is harmed, where E. coli can become parasitic once it enters the urinary tract. So the relationship can change due to the conditions. So normally E. coli is mutualistic to us, But if it occurs where it should not, if it occurs in the urinary tract, it becomes a parasite and it becomes harmful to us. So here we can just see some pretty cool images. This is from your textbook, so you can find them in there, of symbiosis, where you can see commensalism. Here you can see the Staphylococcus bacteria on the skin. Here you can see some mutualism, the E. coli in the large intestine. The E. coli represented here by the purple. Remember, they're these rod-shaped bacteria. And here you can see parasitism. So this is the H1N1 virus that is infecting a host cell, which is in green. So here, the host cell will be detrimentally affected. And looking at some locations of normal microbiota, they pretty much occur throughout our bodies. Again, I think I have mentioned this in the course at some point, where there are typically more microbial cells in you than there are of your cells in yourself. So typically, actually, humans are just all of us are just large petri dishes, large microbiomes. We're just walking microbiomes, essentially. You can view it that way, because they occur in the nose and throat, so the upper respiratory system. They occur in the mouth, the eyes, the skin, the large intestine, and the urinary and genital systems. This includes obviously both sexes, so it's both males and females. So it's not exclusive to one gender. Now, something interesting that I'm sure many of you might not be aware of is that these microbes can actually influence you in some strange ways. So there was an experiment conducted where the microbes present in the gut microbes from two twins were sampled, and they were transferred into identical mice, or similar mice, and then the diet of the mice was changed. It was changed, and it was found that the twin that was obese and overweight, the microbiota it had would still result in increased adipose tissue, so fat tissue in the mouse, even if the mouse was following a low-fat, high-fiber diet. So it has emerged that over the last decade, the gut microbiome plays an important role in the pathogenesis of obesity, where the microbiome profile is altered in obese phenotype, and the causative role of microbiome in obesity is demonstrated in fecal microbiota transplant studies. So again, here, just a breakdown. The microbes were transplanted. The diet of the mice were altered and controlled and was the same, yet the one mouse stayed lean while the other one also became overweight. It's also worth noting that fecal transplants, and with the goal of altering microbial communities within the recipient, have also been used to investigated and used to treat IBD, inflammatory bowel disease in humans, with actually some remarkable success stories where the fecal transplant from healthy patients or healthy donors, I suppose you could call them, resulted in a marked increase in or decrease in the IBD. So it helped alleviate it. Some terminology I want you guys to be aware of. So what is pathology? That is the study of disease. What is etiology? That is the study of a cause of a disease. Important distinction there. Pathogenesis is the development of a disease. Infection is simply defined as the invasion or colonization of the body by pathogens. And then disease is just defined as an abnormal state in which the body is not performing normal functions. Okay, so etiology, again, factors responsible for causing disease. And it's important here to note that some diseases have a very well understood etiology, whereas others are very poorly understood. And some diseases are obviously caused by microbes. These are called infectious diseases, while others are not. There can be diseases caused by nutritional, metabolic, or immunological diseases. And then if diseases are pathogenic and caused by microbes, I want you guys to recall Cox postulates. We discussed them at the start of the course. We're just going to be looking at this very, very quickly because I expect you guys to know all of this by now. And remember that these postulates are used to show that a microbe is the cause of a disease. So essentially, you will take microorganisms and you will isolate them from a dead or an infected animal. It doesn't necessarily have to be a dead animal. And you will colonize them, and you will grow them in pure culture. This is one shortcoming of the method, as you will see in a second. And then you will identify the microbes. Then the microbes will be injected into a healthy animal. Following that, if the disease is reproduced in the second animal, which it has to be to adhere to Cox postulates, the microorganisms will again be isolated from this animal, grown in pure culture, and identified to establish that it was in fact the microbe that led to the disease in the animal. So remember when I said there are some shortcomings, or that there's a problem with the fact that you need to grow these organisms in pure culture. So there are exceptions or shortcomings to Cox postulates. So one of them is that some pathogens can actually cause several disease conditions. This creates a scenario where if you were just to adhere to Cox postulates, you might find differential results where the microbe could produce one disease in one organism and a completely different disease in another organism, which will lead to confusion. Some pathogens can only cause disease in humans, so testing them in mice would not work because the mice would not show symptoms and would not be harmed by the organism. And it's to put it mildly, it's questionable ethics to just be infecting people with pathogens in this way. So that's obviously not done. So that's a bit of, that's actually a big problem. And then the other problem is that some microbes, well, some is as a bit of a misnomer there. I would say the majority of microbes have never been cultured. This is just the fact that, this is driven by the fact that many microbes we don't know what conditions they need, or we cannot mimic the conditions they need in, cultures. Thus, we can't grow them. And that creates a problem for if you want to, adhere to Cox postulates, if you want to follow his postulates where you have to grow this organism in culture. So those are definitely things that you have to be aware of when you start studying infectious disease. So moving on to epidemiology, really pertinent at the moment. So this is the study of where and when diseases occur and of how they are transmitted through populations. So this leads to the work of epidemiologists. We've seen Dr. Fauci, for example, on the news quite often due to the COVID-19 pandemic. So I suppose it's more accurate to call him a pandemiologist, but I'm pretty sure he would work with epidemics in the same way. So these epidemiologists will determine the etiology of a disease. They will identify other important factors concerning the spread of a disease or disease outbreaks. They will develop methods for controlling a disease, and they will assemble data and graphs to outline incidence of a disease. It's really interesting to note here that I think a lot of time these scientists enter periods where they're not as focused in the public eye. Again, we can look at Dr. Fauci, for example, where he only became a real, most people only became aware of his existence due to the entire COVID-19 thing, the outbreak. And I think it's also, these scientists, once an epidemic or a pandemic breaks out, they become absolutely vital. But they also are, learning new things as the information becomes available, which does put them in a strange position where they have to be scientists, yet also be able to communicate with the public and communicate with the media. And that might lead to changing facts because, we don't know everything about it, especially if it's something novel like the coronavirus. We don't know anything, never mind everything. We know nothing about the disease when it first breaks out, or very little. And as facts become more known, they have to be communicated to the public. So I think they have a very, very challenging job, and I would not want that job. Anyway, tangent aside. So epidemiologists can look at things, drawing up graphs to establish, how diseases spread and what causes diseases. So this is chart looking at the colon cancer incidence per 100,000 women on the y-axis, and the per capita daily meat consumption in grams, where you can see this essentially almost this straight relationship, or this relationship forming here of the more meat is consumed, the more likely colon cancer incidence, or the greater colon cancer incidence there appear to be. So there appears to be some correlation there. Then there's also, you can also look at how age, for example, can alter your susceptibility to specific diseases. So here you can look at this graph we have on the slide here. We have age-specific prevalence of respiratory organisms in cystic fibrosis patients, where here is our percentage of patients. So this will obviously range from 0 to 100%, and the age in years. My reason for including this was I just wanted to show you how the organisms or the prevalence of organisms, or the presence of organisms, varies by age, where, you know, at younger ages, the Staphylococcus aureus seems to be the more frequently occurring organism. Yet, you know, if you kind of pass this age threshold of something between 17 and 18, that Pseudomonas aeruginosa becomes the primary microbe that is found in the respiratory patients, or in these cystic fibrosis patients, I should say. So it's interesting how age can also play a role in your susceptibility to different organisms. Important though, I want to note that in that first slide where we looked at the meat consumption and the colon cancer, note how I said correlation and not causation, because if you look at this graph here, it's a very, this will explain why I use that terminology specifically. So here you can see a plot of the number of individuals diagnosed with autism. So this is on this Y2 axis, and then on the Y1 axis we have sales of organic food, and then on the x-axis we have years, and you can see this strong correlation between autism and organic food sales. I mean, just look at that R value and the P value. This is a really, really strong relationship. So the important thing that I want to point out here is organic food sales obviously do not lead to autism. So this graph is a classic example of how a close correlation between the sales of organic foods and the number of autism cases diagnosed does not necessarily mean that it was, you know, autism was caused by organic food sales, or vice versa, that autism caused an increase in organic food sales. So no one has suggested that. So this is a very, very good example to illustrate how correlation does not necessarily mean causation. It's not the same thing. Just because two things are correlated does not mean they are the one causes the other. There was some of you might also be aware of the conspiracy theory at the start of, well, not really at the start, right, recently, about the COVID-19 and the 5G network towers, how people noted that the distribution of these 5G towers seem to be highly correlated with the incidence of COVID-19 outbreaks and people diagnosed with COVID-19. And then that led people to believe, well, this correlation is so strong that it must be the 5G towers causing the COVID-19, which is, of course, completely absurd. But the reason, you know, for that correlation was driven by the fact that was driven by population density. Areas with high population density will, of course, receive more 5G network towers to ensure good network coverage, but it's also very likely that you will see a bunch of COVID-19 outbreaks there because there's a lot of people living there. There's a very high likelihood of disease transmission and spread. So it's definitely, you know, both were caused by the population density, or that population density led to this correlation between these two completely unrelated things, and that fooled people into thinking that 5G towers are giving us COVID, which was not the case. So again, correlation does not mean causation. So moving on to something else that you guys might be aware of. So if we go to South America, we'll be looking at the Zika virus here. So the Zika virus is transmitted by mosquitoes. and it causes, generally in adults, causes very mild symptoms, but when pregnant women are infected by the virus, it can have lifelong consequences for the fetus, including developmental abnormalities like microcephaly, which you can see an example of here. So microcephaly is a condition where a baby's head is much smaller than it should be or can be expected. During pregnancy, a baby's head grows because the brain grows, and microcephaly can occur because a baby's brain has not developed properly during the pregnancy or has stopped growing after birth, which results in a smaller head size. Something that I do want to point out here is also look at the number of cases, the number of, and how that has increased over the years, how there's been this massive spike, or at least there was a massive spike in 2015. Now, this can be driven by multiple factors, but what I do want to point out here is this is one of the perhaps less obvious impacts of a changing climate. If the areas that the vectors for these diseases, so if the areas where these Zika transmitting mosquitoes or Zika carrying mosquitoes can occur and can live in, increases or changes to areas where they previously were absent, this can lead to the spread or the occurrence of this disease in new areas. So, specifically, if you look at something tropical like this and how it's expected that, the areas receiving, these increased temperatures might change or might grow, you can find these Zika vector mosquitoes occurring further to both the south and the north, depending on how you move from the equator. And thus you can potentially have these diseases spreading to countries where they are now currently not found or were originally not found. So not great. Okay, and now we're going to be looking at some of the other portions of epidemiology, specifically dealing with classifying infectious disease. So, I mean, I'm sure most of you have heard the terms epidemic and pandemic and perhaps even endemic. So what does all of this mean? So endemic is essentially just a disease that is always present in a certain population or region. You can almost think of it as a localized disease. An epidemic is a disease that affects many people within a community, population, or region. Typically, epidemics are, once a thing is called an epidemic until it is, anything larger than national scale. So a pandemic then is an epidemic that has spread across a large region, for instance, multiple continents or worldwide, affecting a substantial number of people. So anything nationally or smaller will typically be referred to as an epidemic. Anything internationally or, across at least a couple of different countries will be called a pandemic. You can also classify infectious disease based on whether or not they're communicable. So if you look at something like chickenpox or tuberculosis, they're differentiated from non-communicable diseases such as tetanus. And then you can also look at emerging infectious diseases, something that's new. Again, corona would also fall under this category, but you can look at hemorrhagic diarrhea, which is caused by the E. coli serotype O157H7. This is typically when you've heard about E. coli outbreaks in regions, it's typically caused by this serotype of E. coli. Ebola a couple of years back, it's still around in Africa, but there was that major concerning outbreak a couple of years ago. And then, of course, Zika virus, as we just discussed. These are all examples of emerging infectious diseases. So it's something that's only starting to gain real recognition and becoming a real problem at the moment. Something that I also want you guys to be aware of is when we talk about disease reservoirs, we are referring to the source of a disease. So where is this disease stemming from? This can be either living organisms such as human and animals, the zoonosis or zoonotic diseases. This is where humans or animals, where humans and animals can be the carriers. That's what these living reservoirs is. So just a note here about zoonotic diseases. Just a few notes. Many of the world's deadliest diseases are actually of zoonotic origin. That includes both the plague and rabies. If you know anything about the plague, it just wiped out a huge amount of the European and global populations, over the past, in the past. And then rabies is one of the most deadly diseases known to man. Once you start showing symptoms for rabies, you will very, very likely die. If the disease is transferred to your nervous system, you are very, very likely to die. Not to spook you guys, because it is very, very rare for a virus or a disease to jump over from organisms, to be able to jump between organisms. That includes humans, animals, plants, animals, all of that. It's very, very rare for that to happen, but it can happen. And when it happens, those are typically our worst diseases. And the reason, one of the things that I want you guys to also think about is that typically killing a host is not beneficial for the disease-causing microbes because it prevents its spread. If you are in a human and you kill it very quickly, that will not help your population growth. It will not help your genetic material to be carried forward into the next generation, which is sort of the goal of all life. So, the problem is that these viruses or disease-causing organisms have no sense in what they are currently inhabiting. So if they are currently inhabiting humans, they don't know that. So essentially, if they were originally a disease that made a cow, for example, a little sick, slightly sick, just to aid its spread or to ensure that it can proliferate quite freely and spread throughout the entire cow population. If now that disease is jumped over to humans, it has no idea it is in a human, of course, and it ends up making the human very, very sick and perhaps even killing the human. So that's just a silly example, but it tells you that how these diseases are not really intentionally harmful to the extent of killing people. It's all about spread, but the problem is these zoonotic diseases have no idea what they are currently. So diseases can also, of course, be spread by non-living things. They can active reservoirs. This includes soil, water, and food. Again, with the E. coli outbreaks, a lot of the time it's food that spreads those types of diseases. Transmission of these diseases can be contact, including direct contact and indirect contact, also called fomite, and droplets. This is something that's really relevant now with the coronavirus, with the Rona. We can also have common vehicles, which will essentially be water, food, air, and soil that these organisms can be transmitted by. Or they can also be vectors. Think back to the mosquitoes we discussed, but it can also include other insects or arthropod vectors. This can both be passive or active. We'll be looking at that a bit more here. So looking here at contact transmission, you can have direct contact, where essentially people have to be in direct contact with each other, or it can be fomites, done by fomites, which is essentially an indirect contact within an inanimate object. It's direct contact with an inanimate object. That's what's called fomites, and that constitutes a form of indirect transmission. And then you can also have direct contact through the use of gloves, masks, and face shields. Of course, if you wear someone else's mask, it's not ideal. You can also have transmission occurring via saliva or mucus in coughing or sneezing. This is called droplet transmission. There's a nice example of that. And this brings us quite nicely to the entire mask wearing versus not mask wearing for the Rona. Let's call it a frustration at the moment, because you can still, wherever you go throughout, if you go to a shopping center to do your groceries, you'll still find people not wearing masks correctly, which is very, very annoying, of course. And I think it boils down to a lot of these people not understanding how the masks work, because masks are not really intended to protect you. They're intended to prevent the spread. It boils down to very, very basic physics, where the mask, the covering, will reduce the overall velocity and the momentum of exhaled particles, so you will not be essentially blowing your breath on other people, and thus it will, if you have the Rona, it will not spread to other people because you are not exhaling onto them. You are exhaling to just maybe about here. So unless someone is within this vicinity of you, you won't get sick, or they won't get sick. Okay, and now we're going to be looking at some vehicle transmission. So this is transmission by a medium such as water, food, air. And just a little definition here that I want you guys to be aware of is that airborne transmission refers to pathogens carried by water droplets or dust for a distance greater than 1 meter. I don't know if you guys will recall, but initially it was thought that the corona was not capable of airborne transmission way back when it first emerged. And then after a while, it was realized that no, that very much is possible, that the virus is definitely capable of being transported in these little droplets over a distance of 1 meter or greater. Now, moving on to vector transmission. So a vector-borne disease, and specifically referring here to biological transfer rather than mechanical transfer, is one in which the transmission of infection in a population, the host population, occurs via a second population, the vectors. So yeah, think of this as, of fleas, for example, during the Black Plague or the bubonic plague. So the plague is an infectious disease that is caused by the bacteria Yersinia pestis, which is a zoonotic bacteria, usually found within small mammals and their fleas. It is transmitted between animals through fleas, and humans can then be infected through the bite of infected vector fleas. So again, there are two populations that you should be concerned with here. There is the host population. So in the case of the plague, that was us. And then there's the vector population, which was the fleas that was actually, sitting on these rats and being transported everywhere, typically through shipping, because the rat makes it onto a ship, and the ship goes to a new area, and then you have the rat with the flea on it, the flea actually containing the bacteria. And when this rat either dies or it moves to a new area, the flea jumps onto a human, bites a human, and now the human has the plague. So typically, just a reminder here that these vector-borne diseases, one of the best ways of combating these diseases is simply by limiting contact with the vectors. Of course, this was very, very difficult back in the day, days of the Black Plague, because people had no idea that it was fleas causing this disease. Remember the entire miasma, bad air thing. So it's difficult to keep people away from a thing if they don't know that that thing is really harmful for them. So moving on to nosocomial infections. So nosocomial infections refer to hospital acquired or other healthcare facilities. It doesn't specifically have to be a hospital. And it's specifically found in compromised hosts, because compromised hosts are just simply more susceptible to infection given their immune systems are not functioning properly, which creates a nice opportunity for these opportunistic pathogens. One of the most frequent one is catheter-associated urinary tract infections, which is the most usual type of nosocomial infection globally. It can account for more than 12% of recorded infections. And then we should also be concerned here with the chain of transmission, because nosocomial infections are transmitted by direct contact between staff members and patients and between patients. And then fomites such as catheters, syringes, and respiratory devices can also transmit nosocomial infections. There's a pretty nice example in the show, the series called The Resident. I don't know if you guys watch that or don't watch it, but in the episodes of season 3, episode 19 and 20, there's a pretty cool example where a ventilator that was marked with a unicorn sticker transmits Candida auris between different patients. Why that was such a big problem is C. auris or Candida auris can cause invasive candidiasis, fungonemia in the bloodstream and the central nervous system and the internal organs, which can all become infected. And this fungus is really presents a serious global health threat, which the CDC is concerned about for 3 primary reasons. It is often multi-drug resistant, meaning that it is resistant to multiple antifungal drugs, sometimes even all of the antifungal drugs that are used to treat Candida infections, which is pretty insane. It is difficult to identify with standard laboratory methods, and it can be misidentified in the lab without specific technology. So your smaller institutions will have a very hard time of accurately, identifying the threat before it, spreads. And then obviously, if you misidentify the thing, it can lead to inappropriate management. And it has caused numerous outbreaks in healthcare settings, and for this reason, it is important to quickly identify it. But again, that is very difficult in patients, so that healthcare facilities can take special precautions to stop its spread. So yeah, if you guys have any interest in this type of disease spread, I would highly recommend just watching those episodes of The Resident. Again, season 3, episode 19 and 20. Yeah, it gives you a pretty good idea of how big a problem it can be when these when the spread actually happens, and how big a problem it can really cause if the hospital really doesn't know that it's happening, or the right people doesn't know it's happening, and these fomites are not typically cleaned or sterilized enough. Anyway, so yeah, little tangent aside, I just thought it's interesting to note that, because it is a pretty interesting example. And again, these nosocomial infections are actually more common than you might think, because essentially 5 to 15% of all hospital patients acquire a nosocomial infection. We will be looking at this overlay here again at the end, so I'm not going to be spending too much time at that at the moment. Here you can see some of the microorganisms that are most commonly involved in these nosocomial infections. Notice how it includes quite a few organisms, including the fungi. We just discussed those. And your respirators, which can obviously lead to pneumonia. You can also find surgical wound infections caused by enterococcus. So yeah, it's pretty widespread. Here you can find a table showing you the principal sites of nosocomial infections, with again, your urinary tract infections being most common. I mean, this book, your textbook, states that it's up to 40% of all nosocomial infections. Again, that will vary by source and country and depend on who you're listening to. But take home message, it is the most common one. But if you look at severity, you should definitely look at your respiratory infections. They have a very, very high mortality rate, about anything between 13 to 55%. So that's obviously, a urinary tract infection, although frequent, might not be as severe as these respiratory infections, or obviously something about like a surgical site infection. Yeah, severity is definitely something that you should keep in mind as well, especially you students going on to study medicine. And again, just continuing on with the principal sites of these infections. Again, here, there's a nice little breakdown here at the end. Again, this depends very much whose data you look at. This is data from the CDC, but again, I don't know how updated this textbook is on the newest data that is available, data that are available. But yeah, it gives you a pretty good idea of the breakdown. And then we're going to be chatting here just quickly about how these nosocomial infections result from the interaction of several factors. You need, obviously, the microorganisms present in the hospital environment. You need the compromised hosts, because many of these are opportunistic infections. And then there's, of course, you need to take into account the chain of transmission. And when all these 3 factors overlay, that's where you find your nosocomial infection. So it is notably that the presence of any one of these factors alone is not enough to cause the infection. It is essentially the interaction of all 3 factors that leads to the infections. Okay, and then, yeah, we still have a bit of time left here, so I just thought I'd include this little cartoon at the end here, taking a bit of a stab at the random medical news and how it's reported, at least. So, here the guy will tell you that, coffee can cause depression in children, or in teens, or whatever, twins, or whatever the case might be. So just this, it's very common today in the media that, the media frequently misinterprets, misunderstands, sensationalizes, or misreports scientific studies. I mean, going on with the coffee analogy, how often have we heard that, coffee is good or bad or has no effect or it can now kill you? And it seems to be sort of dependent on, who you listen to and when. So my point in talking about all of this is essentially just that as scientists in training, I want you guys to always question your sources and refer back to the primary literature. So if the original article is included, go refer back to that and make sure it was a peer-reviewed article. And the peer review process and the years of work and expertise is invaluable. It's very common to say that everyone is allowed an opinion, and that might be the case, but not all opinions are equal, and not all of them should be listened to. As a nice example here, if an epidemiologist with a doctorate and years of experience has an opinion about a disease, it has to surely be more to count for more than whatever you hear from a politician, a celebrity, or a less qualified individual. I don't know, perhaps a drunk Uncle Frank or something at a Thanksgiving dinner table. So as scientists, again, you have to be able to assess information critically. You have to be able to question sources and arrive at logical conclusions. It's not all about listening to what's happening on social media or the media or wherever else. Refer back to the primary literature, ask critical questions, think critically, and that will more often than not arrive at the less sensational, less nonsensical answer. You will arrive more often than not at the scientific, the real answer. So yeah, I think it's very important for you guys to be aware of that. Okay, so anyway, tangent aside, let's look at today's review questions. True or false? Nosocomial infections are acquired due to a hospital stay. I mean, this is very, very easy, so I'm not going to spend too much time on it. The answer here is true. True or false? Airborne transmission can include pathogens carried by water droplets. Is this true or is this false? Think back to the slide and think back to the Rona. The answer here is, of course, this is also true. Now, transient microbiota are only present for a few days. Is this true or is this false? I'll give you guys a few seconds. Okay, the answer here is, of course, false. Remember that these transient microbiota can be present for days, weeks, or even a few months. So it's not only just a few days. They are non-permanent microbiota, but the length of their presence is not limited to a couple of days. Moving on to question 4, true or false? Correlation may be indicative of causation. Think carefully and read carefully on this question before you answer. I'm going to give you guys a few seconds. Feel free to pause the video if you need to think about this for some more. The answer here is, of course, true, contrary to what you might think. So in this statement, it's very, I thought I'd include just because it's very, very important that I want you guys to pay attention to little details. What makes the statement true in this case here, the word may, is very, very important in this context. So correlation cannot provide full evidence for causation, but it can definitely be indicative of a causation relationship. So think of this as similar to a first step. Further statistical analysis and testing would be required, but correlation can provide the first indication of causality, especially if the correlation coefficient is large and statistically significant. However, you would need to replicate the conditions, and further testing would be required to establish if it is actually causation. But very, very important here that it may be indicative. If you have a causation relationship between two factors, those two factors would be, or you would expect them to be correlated to each other. So correlation can be indicative of causation, but it isn't the only prerequisite. You need more. You can't just have a correlation, be like, oh, this is causing that. Remember our example that we looked at before, earlier in the lecture, and remember the 5G towers and the incidence of corona that we looked at, or discussed at least. So correlation can be indicative, but it is not the be all end all. So you need more. Okay, guys, that will be it for this one. It's a bit of a shorter lecture today, and then I will see you guys in the next one. Okay, cheers.